Muy buenas noches a todos. Muchas gracias por estar aquí de nuevo. El día de hoy tenemos una presentación increíble, pero antes de empezar tenemos que hablar de cómo van a ser las preguntas. Así que primeramente esta conferencia tiene la opción de ser escuchada en el idioma original, que es en inglés, o puede ser escuchada en español. Lo único que tienen que hacer es en Zoom tienen la opción de interpretation. So ustedes pueden decidir en cuál de las dos. So this conference is going to have interpret. So you can switch between Spanish or English original is the up here. So you in Zoom, you only need to click in the interpretation and switch the idiom you want that. Okay. Sobre cómo serán las preguntas, como las veces anteriores, las pueden hacer en nuestro chat de YouTube o la pueden hacer en el question and answer de Zoom o en el mismo chat. Pueden hacerla durante la presentación. Estas serán llevadas a nuestro presentador. Entonces, vamos a empezar hablándoles un poco sobre quién es Burtom Lim. Bueno, en español, Burtom Lim es un curador asistente de mammalogía en el Departamento de Historia Natural en el RUM. Es nacido y criado en Toronto. Lleva aproximadamente 22 países y contando, realizando trabajos de campo sobre mamíferos. Ha hecho varias disertaciones. Entre estas está sobre la filogenia molecular de los murciélagos de cola cubierta del Nuevo Mundo, familia Emballonurae, su origen, tiempos de divergencia, biogeografía en Centroamérica y Sudamérica, y la evolución de los caracteres morfológicos y conductuales. Él participa en proyectos internacionales como el Barcoding of Life o EVOL, para crear un sistema de referencia genética para la identificación de especies y el descubrimiento de mamíferos. Y no podía dejar de pasar la oportunidad también, por ejemplo, de trabajar con mamíferos como la ballena azul. Entonces, él nos trae una hermosa presentación el día de hoy titulada Cryptic Species of Bats in Dominican Republic and Across the Caribbean based on DNA Barcoding. Les recuerdo a todos que estamos en YouTube y estamos en Zoom. Así que, Professor Burton, thank you so much for staying here, and you can begin when you want.
Okay. Well, thank you very much, and thank you for everybody for joining us today on the Zoom. Yes, so I'll be talking about field work that I've been doing in the Caribbean for about the last 10 years. But I'll be concentrating on the Dominican Republic, looking at sort of the species diversity based on studies that use DNA barcoding to look at the genetic variation that is found in the bats. So most of this work got started actually from in 2009, there was an international mammalogical congress that was in Argentina. And there was a symposium that was looking at DNA barcoding. So from that 2009 symposium, I wrote a paper that basically summarized what was known at that time on the species of mammals. So on in this chart here, on the x-axis are the different orders of mammals that are found in the neotropics. And then all of these bars just show how many of those species have been DNA barcoded. So for example, for rodents, only 12% of the almost 900 species have been barcoded. So the rodents weren't very well represented on the DNA Barcode of Life database. But if you look at bats, Chiroptera, you see that back then, almost half of the species known at that time were actually DNA barcoded. So bats, compared to most of the other mammals, they were actually fairly well represented on the Barcode of Life database. If we look at it from a geographic perspective, you see that the distribution of data points is very, very scattered. So we have quite a bit of localities in Mexico. So the yellow means that there are a few samples, but when you go to orange and you go to red, that means red is the most samples for a particular area. So you see that Mexico is fairly well covered geographically, but there's only a few spots, really more in the Yucatan Peninsula that have a lot of mammal samples that have been DNA barcoded. When you get to South America, you see like in Northern South America, in the Guyanas, so this includes countries like Guyana and Suriname, where I've worked, you see quite a few red dots. So there's quite a few samples from those countries in Northern South America, but you can see like most of South America, like in the Amazon, they're just big, empty, blank spots. So basically, nobody had done any DNA barcoding in those areas. And the rest of South America, again, is also sparsely sampled. When we get to the Caribbean, you see that there, again, back in 2012, there were no DNA barcode samples of mammals in the Greater Antilles or the Western Caribbean, but there are a few that were from the Lesser Antilles, the Eastern Caribbean. So I think those are probably islands like Martinique and Guadeloupe. Yeah, so this sort of served as impetus for me because I had done quite a bit of work in South America and Central America, but I had not done work in the Caribbean. So this was sort of what got me thinking about doing more field work in the Caribbean, because when you talk about working on neotropical mammals, you have to concentrate not only on the mainland, but also on the species that are found in the islands. Okay, just a little bit of background or introduction to Caribbean mammals before we get into the bats. So a lot of this is based on a summary that was done by Liliana Davalos and Sam Turvey in 2012. And in fact, I think this publication came from another symposium in Argentina in 2009 at the International Mammalogical Congress. And I'll be showing you some of their tables of the different species and what islands they're from. But in general, about 79% of the extant or living terrestrial mammals in the Caribbean are bats. So there is this pie diagram over here. So this gives you an idea. So there's 58 species of bats. So that makes up the 79%. There are 13 species of Hutia, which just are these relatively large rodents. And then there's 2 species of Slenodons. So these are like insectivores, but there's only 2 species of those. So you can see the overwhelming majority of mammals in the Caribbean that are extant or living are bats. But then 69% of these bats are endemic or found only in the Caribbean. And then of those endemic bats, 42% of those endemic bats are threatened species. So that just indicates that even though there's not a lot of species in the Caribbean, but the Caribbean is an important area from a conservation perspective because a lot of the species are endemic, but a lot of the species are also endangered species. Okay, so the objectives of the talk I'll be giving today is to look at the genetic diversity of bats in primarily the Dominican Republic, but I'll also be talking about the Caribbean and the neotropics in general, because I want to compare the samples, the populations from Dominican Republic to other islands, but also to mainland populations. And then to also get an overall idea of sort of biogeographic patterns to see are certain species found in different areas, and sort of give explanations on why that might be. So those are basically the three main objectives that I'll be talking about today. Okay, so first I'll talk about the technique that I'm using. So this is something called DNA barcoding. So it actually looks at a mitochondrial gene, the CO1 gene, so cytochrome oxidase subunit 1. So this DNA barcoding methodology was begun, I think, in about maybe 2003. So basically, the people that started it wanted to standardize sort of the species identification, species discovery of animals in the world. So they picked this one segment of the CO1 gene, the mitochondrial gene, but it wasn't just the sequencing aspect of it. It was about collecting specimens. So voucher specimens are associated with the DNA barcodes. And there's a protocol of taking the tissue samples and then the DNA extraction, PCR amplification, and the sequencing. It was also a database. So not just the sequences, but a database that had collection data about the specimens that represented those DNA barcodes. But there are also photographs of the specimens that were collected and that also are cross-referenced to the DNA barcodes. And then they actually had built this platform. So again, all of the records that are online are accessible to the general public. So you can just log on to this website down below here and access all of these specimens. So the specimens that I'm talking about from the Dominican Republic, so those specimens are all on not only this DNA barcode website, but it's also on GenBank. So they're available from a number of different public portals where you can get DNA data from. Okay, so now let me get into the field trip that I did in Dominican Republic in 2015. So I've marked off here on this map. I think there was 11 different points here. So basically I started in Santo Domingo and I just did a clockwise journey around the island. So this was done in about 1 week's time. And we used, fairly standard or typical equipment to catch bats. So we used mist nets, but we took advantage of what we call triple high nets. So these are just long telescoping poles that allow you to stack 3 regular nets on top of each other. So this way you can catch bats a little bit higher up in the air. Yeah, so normally like one net only goes up like maybe about 3 meters off the ground. But this pole allows you to put 3 on top of each other. So you can get up to about 9 meters. So this way you get a better chance of catching the bats that are flying a little bit higher up in the air off the ground. And then there's also a harp trap. So you really can't see it here, but there's fishing line strung from the top of the bar down to the bottom. So usually if it's in a sort of a closed off area, like on a trail or in a cave, like the exit or entrance to a cave, this is a good place for a harp trap. So these are the 2 main pieces of equipment that we use to catch the bats. I had a couple of people helping me. So Olivia Lorero, she at that time was doing her PhD studying the bat genus Molossus, one of the free-tailed bats, which I'll be talking about a little bit later. And this is Nate Upham. His main interests are the caviomorph rodents, so including some of the Hutia that are found on the island. But on this trip, we actually did a separate side project on Hutia and Slendodons. but the main focus of the 2015 trip to Dominican Republic was for bats. Okay, so so even before that and after that, I've done other trips to the Caribbean. So in total, I've circled off in red. So these are 10 different islands that I've surveyed basically over this past decade. So this is basically most of the things I'll be talking about will be based on these field trips, but I've also included other specimen records that I downloaded from GenBank and from other sources. So I do have a few other collections from Cuba, Puerto Rico, and some of the lesser Antillean islands. But a lot of those are just sort of miscellaneous collections. But most of the work that I'll be talking about are based on field trips from 2 to 4 weeks long to these 10 different islands. Okay, so this is just a chart of the different species. So of the 52 species that are found from these 10 different islands, so I think in total there's 58 species of bats, but 6 of them are on islands that I didn't survey. So, you know, for example, like on Cuba, there's a few species that are only found on Cuba. So I wasn't able to get those. Some of them I did get from GenBank, but again, I didn't do any survey work there. So we were able to catch 40 of those 52 species. So the ones that are highlighted in yellow, those are the species and from the islands that we collected or samples from that I'll be talking about in this DNA barcoding analysis. And I've also marked off on the side here on this map, and I've also sort of boxed off, you can just sort of see it. So the first 4 islands are the Greater Antilles, so the Western Caribbean islands, the big islands. And then the next 4 are from the Lesser Antilles, so the smaller islands along the eastern chain of the Caribbean. And then there's 3 islands here, Bonaire, Curacao, and Aruba, that are the continental islands that are just off the coast of Venezuela. So these islands are all within 100 kilometers from the coast of Venezuela. And you can get a bit of information just by looking at this chart. So I've sort of blocked off in black, which you can sort of see, but I've also circled off here, is that there's sort of 3 main regions here. So there is the Greater Antilles group with the large Caribbean islands, so primarily Cuba, Jamaica, Hispaniola, and Puerto Rico. And then the Lesser Antilles, and then the 3 offshore islands that are over here. Okay, so the DNA barcoding data was analyzed phylogenetically. So the CO1 gene included 657 base pairs. So it's not the full CO1 gene, but it's basically the first part of the CO1 gene, and the and all of the sequencing methodology is in this paper, Claire et al. So I put it up here. I'm not sure if you can see it that well, but if not, you can ask me for the reference. But that gives you an idea of how the DNA barcoding sequencing was done. In the analysis I'll be talking about, there were 640 samples that were done of those 40 species. And then the preliminary analyses were neighbor joining trees using what's called the K2P Chimera 2 parameter substitution model. And then there was 400 bootstrap replications and midpoint rooting. So again, I don't want to get too much into the technical details, but if anybody wants to ask, or I can email later on, or you can actually read in this paper how those methods worked. The main reason I use the neighbor joining trees is that they're very, very fast, because a lot of times I'm just doing preliminary analyses, just sort of checking the data. So I don't want to, waste a lot of time, on the computer waiting for something that has a mistake in it and have to redo it. So the good thing about the neighbor joining is that they're very quick. But usually when I go to, publish on some of this work, as I'll talk about in a few more slides, then you will get into analyzing your data more thoroughly with other methods that you've probably heard of, like maximum likelihood, Bayesian analysis, but also looking at other genes, so not just mitochondrial genes, but also nuclear genes. So again, this is basically what I do to get preliminary analyses from the DNA data to start with. Okay, so this is the tree of those 40 species. So I've broken it up into two parts because it wouldn't fit very well on one screen. Yeah, so these are the 40 different species. So basically, I'm not sure how much experience people have with phylogenetic trees, but basically on the horizontal or x-axis is basically genetic diversity, how different things are. So basically, if there's a long branch here to the next one, that means that these things are not that closely related. There's a lot of DNA variation between them. So that's different from, say, between these two species. Yeah, so these two species are in the same genus. So their lines are a little bit shorter, whereas these two species, they're in the same family, but they're in different genera. So these horizontal branches are longer, just meaning that in general, they're more distantly related. And then in the triangle box here, the height gives you an idea of how many samples. So molossus, I can't remember how many, but there's a lot of samples compared to, say, Natalis major. So here, there was only one sample of Natalis major. And here in molossus, there could be maybe 50 samples just because it's higher up. So that's basically the information that you can get from these phylogenetic trees. And then the number is just what's called the bootstrap percentage. So 99% of those 500 replications had all of the individual samples of molossus coming together. So that is fairly good support for this group, for this particular species. Okay, so I'll just give a sort of general overview of sort of the summary statistics that came out of this tree. So the two species that were the most genetically distant apart was this species, Lasiourus minor, and Aerophila bombifrons. So they were 31.2% different. Their DNA was, so that's quite a bit of difference. And so basically, that's measuring sort of the horizontal distance here. Whoops, sorry. Over to here. So basically, the left-hand side is the top of this tree, and then the right-hand side is the bottom of it. So the top here would connect to the bottom here. So they are different by 31% of their DNA. But then on the opposite of this is that the two species that were the most similar are Brachyphylla cavernarum and Brachyphylla pumila. So these two species are only 1.3% different from each other. So that's fairly close as far as genetic distance goes between two species. Okay, so now what I've added on, yeah, so DNA barcoding as far as species identification or discovery goes is that in general, the amount of variation within a species should be a lot less than the variation between two species. Yeah, so obviously things that are different species, they should be very far apart, whereas samples within the same species, their DNA should be very, very similar. Yeah, so these two species were 1.3% different. And those are two different species as we know it now. But even within some of these species, yeah, so the high one here is 4.7%. So that difference is 3 times higher than between these two species. But Macrotus waterhousei is considered one species. So that's a little bit unusual because there seems to be a lot of variation within what we call a species. So usually the ones that I've highlighted here in blue, they have a lot more DNA variation than what I would normally expect. So a DNA barcoding is useful in that it sort of highlights or tells me the species that probably need more work because there's a lot of DNA variation. So now I can go in and try to figure out, why is there so much DNA difference in these things that people call the same species. So these species that are marked off in blue, so these could be potentially cryptic species. So things that superficially look similar, but genetically are quite different. Yeah, so basically for the rest of my talk, I will be giving more detail about these 5 different species. Yeah, so basically everything else, all these other species are more or less, they had the expected amount of DNA variation. So I'm not going to talk too much about those. So I'm just talking about the ones that have a higher than expected amount of DNA variation. Yeah, so the first one I'll talk about is molossus. So within this group, the average DNA variation between 2 samples was 2.4%. So that's the average genetic or DNA variation. So molossus is sort of the easy one because just recently there were a couple of papers published that resolved the taxonomic status of this species group. So again, based on that original table that I showed you earlier on, so that was so back in 2012 when Davalos and Turvey made that table of species. There was only one species of molossus that was found in the Caribbean. But based on an initial study by Livia, who did her PhD on the genus Molossus, looking at the systematics and taxonomy. Yeah, so what I boxed off in red here, so originally these were considered one species, but you can see that they're not even close to each other. So they're actually quite different, even though people call them, the same species. So this was based on more traditional mitochondrial and nuclear genes. So 5 genes, this is the combined data set. So for her PhD, she used another method using next generation sequencing. So this looks at what are called SNPs, single nucleotide polymorphisms. Yeah, so you get a lot of data using this method, but as you can see here, the 2 groups, the 2 boxes still come out separate. So what normally is considered molossus, that species, is the species that's found basically from the mainland, from Central America, South America, but also into the Lesser Antilles. But then the 2 species down here in this red box, One of them is Molossus virilli. So that is the name that is now used for molossus that's found in Dominican Republic. And then the name Molossus milleri is the name that applies to the molossus bats that are found in Jamaica. Yeah, so this has happened. So these two papers came out last year and this year. So both sort of the typical sequencing methods using individual genes came out with the same result as using a genomic approach. So again, both of these papers corroborated the fact that these two species are cryptic species of molossus. So they already had names, so we didn't have to give new names to them. So old names were elevated to the level of species. Okay, so this is looking at Piptescus fuscus. The English common name is the big brown bat. Very exciting name. And again, we have a tree of the different species of Piptescus. Again, looking at DNA barcoding. So here, the scale here, that's 2% there. So basically, yeah, so there's about maybe 6% average. So that's, again, relatively high. Usually when you get, over, 3 or more % difference, then I start wondering, what's going on. So this here is about 6%, and that's only the difference between Dominican Republic and Cayman Brac. So Cayman Brac is one of the 3 islands that's part of the Cayman Islands. So just west of Jamaica, south of Cuba. So there's 6% difference between the populations on Cayman Brac Island and on Hispaniola. But again, if you look at the big brown bat from Canada and the US, you see there's even more difference between that. So there's a lot of genetic variation going on in what people call, the one species of big brown bat. So again, more study needs to be done, but there's a potential that there might be cryptic species here. But again, there are already older names that are available. So the one that's found in Dominican Republic, if it was elevated species, it would be given the name Hispanioli. And then the one from Cayman Brac, I'm not really sure what name to give it. So that's going to take a little bit more research. There's no names from the Cayman Islands, but there are potential names from Cuba that might apply to the population from the Cayman Islands. But you would need samples from Cuba to see if they're the same or different from the samples from Cayman Brac to figure out what name they are. But again, the DNA barcoding indicates that there's something unusual going on here that may require more work to see if they really are true cryptic species. Okay, so this one is looking at a species of nectar feeding bat. So this is a Monophilus redmani. So again, it's considered one species. So we have samples from Dominican Republic and from Jamaica, but you can see there's 8%. So 8% again, for DNA barcoding, that's pretty high. There's another species, Monophilus plethodon from the Lesser Antilles. So that's even more different. So that's 14% different. And that one is considered a different species. But the species from the Dominican Republic and Jamaica the specimens are called the same thing, even though there's 8% difference. This is what the bat looks like on Jamaica, and this is what the bat looks like in the Dominican Republic. So I can sort of see some differences. Now whether that is common or regular differences throughout the population, that again will need more study, but definitely from a genetic perspective, there's quite a bit of difference, and there also might be some morphological differences. So again, just an indication that that more work needs to be done, beyond DNA barcoding. But there are names available for these two, so there probably won't have to be a new name attached to it. So the one from Jamaica is where the species was originally described, so it would get the Redmond eye subspecies name, but then the one from Dominican Republic has a separate name, but if it is considered a different species, it would use this name, and that would be elevated to species. Okay. Okay, and then the last two examples have a slightly different story. So this is one of the mustache bats. So it gets its name because you can just sort of see some very fine hair sticking out from around its mouth. So that's where the name mustache comes from, at least in English. And what's interesting here is that even though we got samples from the Dominican Republic, there was a bit of difference between the north part of the island and the south part of the island. So I've drawn a red line across this part here that sort of separates these two populations here. Yeah, so I think between these two populations in the Dominican Republic, I think that's about maybe 4% difference. So again, that's pretty high, from one island. So like the last example I showed you for Monophyllus, that was between two islands. But here, this is 4% within one island. So again, very unusual. That would require more research to look into what's going on there. And this is the last example I'll show you here. So this is a species, Macrodus waterhousei. So again, a lot of different, like 7%, again, between the north part of the island and the south part of the island. So again, that same break here. Yeah, so there's something happening in this particular area within this particular species. So it's considered one species, even though there's quite a bit of genetic difference. And here are pictures of what these two look like. But this break is not, people have known about sort of a difference between the north part of Hispaniola and the south part of Hispaniola, the island. Yeah, so there's a paper that came out in 2012, again, looking at Hutia. So those are the big rodents. And again, they found a break between the north part and the south part of the island. And then they also found another break in the western part of Haiti. So I didn't do any sampling on the western part of Hispaniola. So all the information I have is just for Dominican Republic. But as you can see, like even within one species, there's quite a bit of sort of phylogeographic or biogeographic difference within one species on one island. But this same pattern is similar to those last two examples of bats I gave you. And there's also examples in birds also. So historically something had acted as a barrier between the north and south part of the island. So basically saying that there needs to be a little bit more work that needs to be done. Okay, so I'll just quickly summarize here. So sort of the three sort of takeaway points you can get from my talk is that in general, there's sort of three biogeographic regions that are found in the Caribbean, the Greater Antilles, the Lesser Antilles, and then the offshore continental islands just off of Venezuela. There's a bit of sort of dispersal across these regions. But in general, there are distinct groups of bats that are only found in these three. Some species are found throughout all three of these regions. In fact, I think there's only one species that's found in all three of these regions. But more than half of the species are found only in one of these three regions. So in general, there's three biogeographic regions in the Caribbean. So I've given you 5 examples of potential cryptic species of bats, and they all happen to be in Dominican Republic. But remember, I only sampled 40 of the 52 species. And in fact, there are 58 species. So if you go to the other islands I didn't sample, I think there's a possibility of making even more discoveries on other islands in the Caribbean. And then the last sort of point I wanted to summarize is that there were 2 species of bats that had unusual break, within the island of Hispaniola. There's a population that was distinct in the south and a population that was distinct in the north, which is also found in rodents, but also birds. So from a biogeographic perspective, this is something that deserves more research effort into this particular sort of phylogeographic break in the island. Okay, so I'd like to end off by thanking the Ministry of the Environment and Natural Resources in the Dominican Republic. So they authorized the research permits, but also issued export permits for specimens and tissue samples. We collaborated with the Natural History Museum in Santo Domingo. So thanks to them, and also thanks to Miguel, who also helped out on the trip, but also who invited me to give this presentation. So thank you. Most of the funding for the field trip in 2015 came from a grant I got from the Royal Ontario Museum, the Shad Conservation Grant. Okay, so that's the end of my talk. I hope you enjoyed it, and I will be available for any questions that anybody might have. Thank you.
Thank you, Dean. We have some questions. So the first is from Hugo Mantilla. He said, "Dear Burton, you mentioned that are several clades representing species that have more variation than expected in a single species, maybe representing cryptic species. If you have to choose between those 2 potential explanations for the accumulation of variation, demographic characteristic of the species, group size, mating system, and time of isolation, which one you will choose?"
Well, I think some might be a little bit easier to decide, what to do. Yeah, so for example, the Monophylla redmani, so that was a high species variation between 2 islands. So obviously, the islands can act as a barrier, to separate populations. Yeah, so that one might be a little bit easier to explain, and might be, a reason, to elevate elevated to a new species, a different species. But the examples, the two examples from Hispaniola between the north and the south, so that might be a little bit more difficult to explain because obviously there's no barrier now, but maybe historically there was a barrier because I think that red line I had was a depression. So maybe during a time of higher sea levels, I think water had separated the north and south part of Hispaniola. Now, whether it was actually like a sea channel or whether it was just, sort of a saltwater lake, that I'm not really sure. But yeah, so that explanation might need a little bit more study because, that barrier is not there anymore. So the question is, was that barrier long enough to have separated two species? Or was the barrier not long enough. So now there's gene flow between the populations. So what we're just seeing is just a sort of a molecular signature of something that happened in the past, even though they might still be one species now.
Okay, thank you.
I hope that answered this question.
Yeah. The next question is for Paul Velasco. Said, hi, Burton. This is Paul Velasco. Great talk. Do you have and did you analyze acoustic information for the bats that present the genetic break in the island?
I did. I think that might have been, so that was back in 2015. So that's when I just started to do acoustic monitoring. So I did have a bat recorder that I was putting out, but it sort of broke down in the middle of the trip. So we do have some information, but I'm not sure. I'll have to go back and double check it. So it's possible that I do have recordings that might give more like a separate data set, like acoustic data set to support what the genetic data is. But that's something, again, that I'll have to work on along with the morphological differences. So we also saved like museum voucher specimens. So we can also, measure specimens and look at different morphological characteristics. So that is still ongoing work, part of this project.
Thank you. The next question is from Catherine Pino. Said, this differentiation of the COI gene may be due to a population structure greater than different species. Considering that only mitochondrial genes have some issues like maternal inheritance, recombination, and inconsistent mutation rate?
Yeah, so as I sort of mentioned at the beginning, yeah, so for me, I use DNA barcoding more like a taxonomic tool to sort of help me. Yeah, so in general, most of the times, people, when they identify species, they look at the morphological differences, the color of the fur, or, if it has, stripes on its face, how many teeth and stuff like that. But sometimes things look similar. So I use DNA barcoding as just sort of the first step in sort of identifying species, but also trying to figure out whether they are the same or different species. So as was done with molossus, so originally DNA barcoding told us that, the populations from the Caribbean were genetically quite different from the mainland. So that's when Livia started using other data sets, such as the SNP data set. Yeah, so for me, I use the DNA barcoding to sort of help me decide what groups of bats need more study.
Okay, thank you. Another question for Álvaro Benitez says, thank you so much for your presentation. It's about N. molossus. Where is the distribution and is or not occurring in Dominican Republic?
Sorry, was that molossus you said? The species? Yeah. Yeah, so in the Dominican Republic, on the island of Hispaniola, the molossus is, the new name is Molossus virili. On the island of Jamaica, the new name is Molossus milleri. So molossus, the old name, is found in Central America, South America, and then into the Lesser Antilles. And then molossus is also found on the continental islands. So on Aruba, Curacao, Bonaire, off the coast of Venezuela, that is also molossus. So at this point, there are two new or added species from Jamaica and from Dominican Republic.
Okay. Another question is for Aida Otolara Ardila. Do you know what are the ecological or geographical factors that could drive the phylogeography differences in the two bat species between the south and north in the Hispaniola?
I think in general, someone correct me that actually lives in Dominican Republic on the island. But I found the southern part was seemed to be a little bit more drier, and then the northern part seemed to be a little bit more humid or more wet, because like basically right in the middle of the island is sort of that mountainous region. So that I think has an effect on the climate. Probably not only between the east and the west, but also the north and the south. So I think sort of the central mountainous area might be something that affects the ecology or the habitat on the island.
Okay. Another question from Jacqueline Salazar. Jackie Miller here for the RON. In what direction do you see this research developing?
Well, I think it's more of a broader, so so to me, it's not just, the Dominican Republic, it's not just the Caribbean, it's the whole neotropical region, so Central America, South America, and the Caribbean island. So in general, the species in the neotropics are, different. They have a different faunal composition than, say, from North America, and totally different, from, say, the Old World in Africa and Europe. So I just want to broaden sort of the analysis or the research across, as many different parts of the neotropics. And of course, the Caribbean has a lot of different islands, and I haven't been to all of them. But basically trying to get as good a geographic sample as possible.
Another question for Hugo Mantilla. He said, you guys have conducted some sort of analysis on population expansion or contraction mismatch. For those species, for which you guys more data? If yes, what they said?
Sorry, could you repeat that again, please?
Yes, of course. You guys have conducted some sort of analysis on population expansion or contraction mismatch. For those species for which you guys more data? If yes, what they say?
I think I think we always need more data. because again, I only sampled 10 different islands, but again, throughout the Caribbean, there's a lot more. And the biggest island, Cuba, we don't essentially have any samples from. So again, these are all just sort of preliminary results. So basically, when we do the DNA barcoding, and papers are published, they're all made available. So this way, other people can use them. So it doesn't have to be me, that continues studying these more. Anybody can go on to GenBank and download, the barcode sequences and add them, perhaps to some samples they have. So I think there's a lot of more and different studies that can be done, and other people are more than welcome to study these animals because the data is freely available on the internet.
Okay, we have 2 questions, so I need a little time for translating. It's from Miluska Ramos Sanchez. She said, "Thank you so much for your presentation." And the question is 2 questions said, how you can explain differences between species with low percentage for genetic distance, like Brachiophila with 1.30 percentage. If you compare with other species with more percentage. I didn't understand exactly the question. And the other is, what factors can be cause the distance between 2 populations, for example, Pteronotus? Would you explain in the beginning about the north and the south of the island?
Okay, so for the first question, for Brachiophila, so my talk was titled Cryptic Species, but it's possible it could be the other way. There might be things that people call 2 different species that maybe might end up being 1 species. So that's the opposite of cryptic species. I don't know what they call that, but so that's a possibility. So again, I think that also needs more research because based on that low percentage, it's a possibility that they're not 2 species, but in fact only 1 species. So again, more work and more samples, because I think for that species between I think it was Pumila and Cavinarum, I think they might have just been from 2 different islands. So again, it would be good to sample, the islands in the middle that we don't have any specimens from. And then the second question about the Pteronotus, I think they were asking about how could samples be different genetically. So again, any type of type of geographic barrier, or maybe even ecological barrier. So that one question about where I gave the answer about potentially different habitats, dry forest and wet forest. So things can also speciate, if they're isolated, in different habitat types. So if 2 populations, don't come in contact because one only lives in sort of dry open forest, or one lives in only the wet rainforest. Again, time is very important. So over a long period of time, if they remain isolated, and then they can diverge into 2 different species. But again, the example of, again, with Monophilus, when one was on the island of Jamaica, one is in the Dominican Republic, then obviously they're separated by ocean, so that can isolate things. When it's on the same island, it becomes a little bit trickier to try to explain that. So some of the explanations might be ecological, or there might be a barrier, like an old oceanic seaway that separated the south and the north.
Okay, another question is for Carlos Emilio Molano Paternina. What kind of genes can be, what kind of support genes can be, okay, ¿qué genes pueden ser respaldo? Okay, what genes can be support for mitochondrial genes, and what kind of analysis can help for the SNP? Sorry, it's in Spanish. I'm trying to translate. Do you understand?
Yeah. Well, the
good question.
yeah. So yeah. Okay. I think I know the question that they're asking.
I'm going to cut the question because it's two. What kind of genes can be support for mitochondrial genes? And the other is what kind of analysis can help for the SNP? I think it's two.
Okay.
Yeah.
Yeah. So if you have a mitochondrial gene, usually if you want to expand your study, with more molecular work, you would probably want to go to a nuclear gene. Yeah, so in general, I think you only really need one mitochondrial gene, just because all the genes on the mitochondrion are all linked. So in general, one gene from the mitochondrial DNA will more or less tell the same story. So if you want to get support or to refute, the mitochondrial gene, if you went to nuclear genes, so now that is, biparental inheritance, basically one from each parent, whereas the mitochondrial gene is just from mother to mother. So in general, when you do more studies, it's good to have a mitochondrial gene and nuclear genes just to see if they're telling the same story. Yeah, so if I wanted to take any of these particular stories that I had said, I would probably try to sequence, several nuclear genes. Then the second question was it how to analyze the SNPs? Is that, I think that's what they were asking. Yeah, so the SNPs, you can get a lot of data out of it. So I think when Olivia got the SNP data set back, I think there was like maybe 300,000 pieces of data. Like, so SNPs is a single nucleotide polymorphism, like one DNA change. I think she had like over 30,000 of them. But the analysis, again, can be a little bit tricky because you have to filter out, because not all of that information is useful information. Sometimes it just, noisy information that doesn't, that won't lead you to like like the wrong answer. So a lot of times you have to do different filtering techniques to get down to a core group of sort of phylogenetically informative characters, SNPs. So I think, again, it's a little bit tricky to do, to figure out, when to stop, the filtering method before you start analyzing the data. But again, it really depends on, the group you're working on. And if there's a lot of similarity, then you might have to, just do more analysis, more work, to get to like a reasonable answer.
Okay, we don't have more questions, but someone say hello for you for wildlife. So Yeah. For you too, someone say hello, nice to stay here. It's wildlife. Professor, thank you so much for your conference. It was so great. So a lot of questions about the important information you give to us. And we're going to finish with your presentation. And we come in 15 minutes with Alexa Sadier. In 15 minutes. So again, thank you so much. We appreciate every work you're doing, not only in Dominican Republic, also in the Caribbean and the world. We expecting so much papers in the future, not only for Dominican bats or Caribbean bats, but also for more species you can give us. And we appreciate your time and your knowledge for sharing us. Thank you so much, and we hope to have you in another moment in that pandemic world right now.
Yeah,
So,
thank you for all of the organizing that was done, and thank you for everybody out there in the Zoom world. Thank you.
Thank you so much. Para nuestra audiencia, vamos a regresar en unos 14 minutos, así que por favor, vuelvan a conectarse o por YouTube o por Zoom con el mismo link que les fue enviado a sus correos. Ahora mismo nos vamos a organizar con nuestra presentadora, Alexa Sadier.